I'm the awesome tutor, and today we're going to be looking at cell division. First, we're going to look at cell division and growth. Now, why do you need new cells? New cells are obviously needed for an organism to grow. So growth is the first thing. Next, you also need new cells to replace the worn out cells. When cells become worn out, they die and so do not function properly, and you need new cells to replace them. You also need new cells to replace the damaged cells or damaged tissue. You know, when you cut your skin, those skin cells have now been destroyed, and you need new cells to replace them. That's how your cut heals. Obviously, first you've got the platelets and other things from your defense mechanisms, but that's module 1. Now, cells that do the same job must have the same genetic material as the original cell. The original cell, to produce identical copies, must divide, and it divides to produce two identical cells that have the same genetic material as itself. And because they have the same genetic material, they're going to be doing the same job. Now, where do we find these instructions to make new cells? The instructions for making new cells are found in your genes. And a gene is a small packet of information that controls your characteristics. Now also, we've got alleles. What are alleles? Alleles are different forms of genes. Depending on which allele is dominant and which allele is recessive, that will control your phenotype. So alleles, different forms of genes. Now in this video, I will be going over, there are two types of cell division. There's mitosis and there's meiosis. Now we're going to talk about mitosis. Okay, so mitosis. Mitosis is a form of cell division. And it is cell division that produces two identical cells, as shown in this beautifully drawn diagram. Now first you start with a normal cell. Say this is a body cell. A normal cell with a complete set of chromosomes. You've got two pairs of chromosomes here. Now in a human body cell, obviously there are 46 chromosomes, 23 pairs. But I cannot draw 46 chromosomes on this small diagram. So just imagine that this is a full set of genes, a full set of chromosomes. So the normal cell has a full set of chromosomes. Then, before the cell divides, it duplicates the chromosomes. So then duplicates the chromosomes identically. Identical duplication. Why? Because when it divides, it produces two cells which are identical because they have the same chromosomes, because they were identically duplicated. So this happens right before it divides. Then it actually divides. It divides into two daughter cells with identical genetic information. So half of the set of genes here goes there, and then the other half goes there. And that's how they have identical chromosomes, because here they're identically duplicated, so when it divides, it has the same chromosomes as the first, as the original. Now, before we talk about meiosis, let's talk about differentiation. Differentiation is the specialization of cells for a specific function. Now, in the early development of the embryo, cells are unspecialized. And we call these unspecialized cells stem cells. And these stem cells can become any type of cell that is needed. And when it does this, we say it specializes. And specialization is when a cell adapts to perform a particular function, a specific function. And for this specialization to occur, some genes will be switched on, and some genes will be switched off. Now, unlike animal cells, differentiation in plant cells can happen all through their life. But in animal cells, like us, differentiation only happens in the early stage of the embryo. But in plant cells, differentiation takes place all through their life. Now, mature adult animal cells, their cell division is mainly restricted by mitosis. Now, undifferentiated plant cells, where do you find them? Where are they formed? Where are they created? They are formed in the active regions of the roots or even the stems of the plants. And in these active regions of the roots and the stems, mitosis is continuous. The plant stem is continuously growing. The plant's roots are continuously growing. And in the right conditions, the cells in these active regions can become unspecialized. They can go backwards and unspecialize. And this helps us to create huge numbers of identical plant clones. So say you cut off the root or a stem of a plant, and then you give it the right conditions. It will unspecialize and undergo mitosis many times to create an identical clone of the original. However, in animal differentiation in animal cells is actually permanent. We cannot go back and unspecialize these cells that have already gone through differentiation. And so animal clones can therefore only be made by cloning the embryo at its early development stage, when the cells are unspecialized. That is the only way you can create animal clones, because animal differentiation is permanent, cannot go back, and it happens only in the early stage of the embryo. And unlike animals, in plant cells, specialization differentiation takes place all through the life, and they can unspecialize in the right conditions. However, you can make an embryo from an adult cell, but still, it's restricted. Now we're going to talk about meiosis. So, cell division in sexual reproduction. Mitosis takes place continuously, and yet meiosis takes place only in the reproductive organs. And in meiosis, the number of chromosomes in each cell is reduced to half the original set. So the number of chromosomes are halved, or half. Okay. And this results in the formation of sex cells, or gametes. Sex cells are called gametes. So they might ask you in your exam, what are gametes? They're sex cells. So now, what is the proper definition of meiosis? If they ask you for it in your exam, what will you write? Meiosis, first of all, cell division, where chromosome number is reduced by half, is reduced to half, resulting in the formation of gametes. And that should give you full marks on that question, should it come. And now the next part is actually required only of the higher tier students, which is the actual process of cell division in meiosis. But then I would also talk about something that involves all students, so you could skip the next part, or you could keep watching just out of interest. Okay, so in meiosis, you start out with a regular cell in the reproductive organs. And then this regular cell duplicates its chromosomes. So identical duplication of chromosomes. And then after it does that, it divides into two to form two daughter cells with, of course, the identical copies. So divides into two. Wait a minute, this is just mitosis. But there's one more stage in the cell division of meiosis which differentiates it from mitosis. The cells divide again. And because they divide again, they do not contain the full set of chromosomes as the original. They contain half. And so they contain only a single set of chromosomes. And as you can see, 4 gametes are formed, 4 sex cells, each with a single set of chromosomes. And however, each set of chromosomes is in a different combination. So they have a random mixture of chromosomes from the original. And this helps to allow variety, genetic variation. And so now they only have half the set of chromosomes. And when they fertilize with the other half from the other sex cell of the other gender, they're going to fuse and combine. So half plus half will then give you a full set of chromosomes. And that's how even more variety is introduced. Next. Okay, so during fertilization, more variety is added, as I just said before. And each gamete has a single set of chromosomes, as we just established. And so when the cells, the two gametes fuse and they join in fertilization, a single new cell is formed. And this single new cell now has a full set of genes, because the two gametes, each of them had a single set, half the set. 23 from one of the gametes plus 23 chromosomes from the other gamete is going to give you 46 chromosomes, which is the full set of chromosomes in a human cell. And the combination of genes of the fertilized cell is now going to be unique. And because of the immense variety that has been added and the completely random mixture of chromosomes and genes, it is going to be unique. And then, once the new single cell is formed with a full set of genes, it starts to undergo mitosis. And that is how an embryo is formed, and then a fetus after specialization, and blah 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 blah. Next. Okay, so let's just give an overview of asexual reproduction and sexual reproduction. In asexual reproduction, the offspring that are produced is due to mitosis. Because remember, mitosis produces identical cells. And so the mitosis occurs from the parent cells, and that's what produces the identical offspring. So the offspring are identical to the parents in asexual reproduction. In sexual reproduction, the gametes are produced because of meiosis from the reproductive organs of the parents. And so in asexual reproduction, the chromosomes and the genes are exactly the same as the parent cell, or the parents themselves. And in sexual reproduction, as we established, each gamete is different. So it's going to be different from the parent. And so which type of reproduction do you think will have genetic variation? It's not going to be asexual reproduction. There is no variation there. But there is variation in sexual reproduction. Because different combination of chromosomes, gametes produced, and so they are genetically different. Asexual reproduction, because of mitosis, identically copied cells, exactly the same as the parent, no genetic variation. And now let's move on to the exam question. Okay, so we have several exam questions on what we just talked about. So the first one, during in vitro fertilization treatment, each fertilized egg cell forms an embryo by cell division. Name this type of cell division. So it's either going to be mitosis or meiosis. Now you might think, because of the word fertilized and the word egg, you're going to think meiosis. But it's not meiosis, it's mitosis. Meiosis is the formation of gametes. Mitosis is identical duplication of cells. It says the fertilized egg cell. So the egg cell has been fertilized. That means the gamete and the gamete has already fused. So now it has a single set of chromosomes. And then after this, it begins to divide by mitosis. So you need to know the difference between mitosis and meiosis, and definitely read the question properly in the exam. You have to know how to spell mitosis and you have to know how to spell meiosis, because they are very similar in terms of their letters. So if you spell it wrong or your handwriting is illegible and the examiner sees it as the other type of cell division, you're going to lose the mark. So make sure to spell them correctly and make sure to write them clearly. How many cells will there be in an embryo after the fertilized egg cell has divided 3 times? Well, now that you know it's mitosis, mitosis produces 2 identical cells. So if it does that 3 times, 2 times 3 is 6, right? No, that is a common mistake. If you have this cell, it's going to divide into 2, right? So now there's 2. Now each of those 2 cells are now going to divide again. So this is going to be that, and then that's going to be that. And then it's going to divide a 3rd time, so then this will divide into 2, this will divide into 2, this will divide into 2, and this will divide into 2. So it's not the egg cell, divides into 2 3 times, it's the number of cells doubled 3 times. So it's not 2 times 3, it's 2 to the power of 3, so it's 8. Of course, you could draw a small diagram to figure it out, which actually I would do, just to make sure. Now, the genetic material of the embryo is made up of large molecules of a chemical substance. Name this chemical substance. This is just knowledge, no explanation. It's DNA, or deoxyribonucleic acid. If you're going to use that long word, then you have to spell it correctly. So just write DNA. Describe where this substance is found in a human cell. 2 marks. Where is DNA found? To do that, we need to know the structure. So first of all, it's genes. Genes are found in your DNA. DNA is found in your chromosomes. And chromosomes are found in the nucleus of the cell. So a segment of DNA is a gene. But now I want you to find out where the DNA is found. DNA found in the chromosomes, found in the nucleus. So if you write in the chromosomes, in the nucleus, there are your 2 marks. 1 mark for chromosome, 1 mark for nucleus. That's it. So, bye.